So we're going to continue on here, and we're going to look at the next set of tissue types, which is muscles. And so we're going to look at muscle. Muscle is generally characterized as being what's referred to as an excitable tissue. That means that when the membrane potential itself changes, it will go about doing a functional and structural change to the tissue itself. In this case here, it's going to be structural through a process known as being contractile. That is, it will be able to shorten actively the length of the muscle or the cell itself. It is elastic, which means that it can stretch past its uh, standard resting length and then return back to its initial resting length. We look at muscle in terms of its regionalization, in which we have three distinct classes of muscle based off of the regions that we see it. If it's found in the heart, it's referenced as being cardiac. If it's found attached to skeletal tissue, it's referred to as skeletal muscle. Any other muscle is referred to as smooth muscle. In this classification, we have other classifications that we look at. The classifications that we look at is based off of the presence of a transverse tubule, which leads to striation patterns within the muscle tissue itself. Cardiac and skeletal muscle is referenced as being striated, meaning it has transverse tubules. Smooth muscle lacks transverse tubules, hence its reference as being smooth. And so if we look at this quick image here and we'll look at it in more detail and we look at the slides in lab, you can notice the striations within the skeletal and cardiac muscle, and you do not see those same striations within the smooth muscle. So let's take a look at the muscle characteristics themselves based off of the three types. And we'll start with skeletal muscle. Skeletal muscle is referenced as being a polynucleated cell. That means it has multiple nuclei. These nuclei are going to be peripherally located. They're peripherally located as the bulk of the cytoplasm itself, the bulk of the cell structure itself, is made up of cylindrical uh, contractile tissues. Skeletal muscle itself is also referred to as being amitotic, which means that once it's mature, it does not replicate and produce new cells. However, there are what are referred to as satellite cells within skeletal muscle, which will undergo replications, allowing for a process of growth known as hypertrophication to take place. Skeletal muscle is the voluntary of the muscle types, which means that it needs to be externally recruited in order to contract. And in this case here, it is going to be neurologically controlled. That neurological control can be modified by based on what's referred to as a stretch reflex, a preloading, or what's referred to as an ease of depolarization, which we'll get to when we talk about actual contractions of muscles, not in the anatomy class, but in the physiology class. And so if we look at the skeletal muscle itself, what you'll notice is there is the peripheral nucleus filled with the bulk of the sarcoplasm, the cytoplasm for muscle, being the myofibrils, the actual proteins that will contract. Here indicated as being thick and thin filaments. The thick filaments are the protein myosin, and the thin filaments is the protein actin. Surrounding the cell itself is the sarcoplasm. The sarcoplasm has openings or pores within it. These pores lead into the transverse tubule and allow for depolarization to take place across the length of the muscle itself. The length of the muscle is going to be the length of what's referred to as the fascicle of the muscle, and we'll get more detail when we talk about muscle anatomy itself. If we look at the histological image of the skeletal muscle, notice the striations running vertically through the muscle fiber itself. Notice the peripheral nuclei, and then you'll see all these kind of other purple dots within the muscle. All of those other purple dots are the mitochondria. The amount of mitochondria that we see within the muscle tissue is going to dictate whether it is an aerobic tissue or oxidative tissue, or whether that tissue is going to be an anaerobic tissue or glycolytic tissue. Smooth muscle, on the other hand, does not have multiple nuclei. It has a single central nucleus within the cell itself. There's no distinct form to the smooth muscle, which is why we reference it as being fusiformed. There's no T-tubules, there's no transverse tubules, and so we don't see any type of striation pattern. This is typically involuntary, meaning that we cannot cognitively cause the smooth muscle to contract. However, it is neurologically modified based off of autonomic nervous system signals. It will respond to stretch reflexes as the muscle itself elongates. It triggers a contraction to get back to resting length. And it's modified in a different contraction pattern than what we see with the other types of muscle. And so here is the smooth muscle itself. And so if we look at the smooth muscle, notice that we have this kind of central nucleus, and then we have these kind of irregularly shaped cells themselves. When it contracts, what it does is it has these bands within them known as the dense bodies and intermediate fibers connecting them. When the depolarization takes place within the smooth muscle, the actin and the myosin contract in such a way so that we go from having this kind of elongated wormy shape to kind of this squished up wormy shape of a cell. So it goes from this elongated cell to this shrunken cell, but it's very haphazard in terms of the way in which it contracts. And so the way in which it contracts is based off of the movement of calcium into the cell itself, leading to additional calcium being released, activating an enzyme known as a calcium calmodulin, which initiates a secondary activation sequence, which goes about causing phosphorylation on the myosin, leading to the contraction of the muscle itself. And so you'll notice that we have these rings of muscle around the esophagus in the slide. It's the ring of muscle that is going to control the contraction wave that allows for food to move from the mouth through the esophagus into the stomach. The next set of muscle tissue is cardiac muscle. Cardiac muscle, just like smooth muscle, has a central single nucleus, or centrally mononucleated. It has transverse tubules, just like with the skeletal muscle, so it's going to look striated. Even though the cells themselves are individualized, they are interconnected by what's referred to as an intercalated disc. This allows for a coordinated contraction to take place amongst the cells themselves. They are bifurcated or Y-shaped, and the shape of the Y is actually in such a pattern that they are inverted to each other. So the top of a Y is going to match up with a top of another Y in an opposite pattern. And then they overlap each other at varying angles, so that when it's happening, we get this mesh of cells that all line up at different angles to each other, so that when the heart expands to allow for an increased volume within the atria or within the ventricle, we don't get rupturing of the muscle itself. What's interesting about muscle in the cardiac sense here is that it is a lipid dependent tissue in terms of its fuel source, and it's an aerobic obligated tissue, meaning that it's going to be dependent upon aerobic metabolism, and it needs a constant perfusion in order to allow that to take place, meaning that it has to have constant rate of blood flow heading around. Aerobic obligated tissues within the body have a maximum of 5 minutes of lifespan before they start becoming necrotic and infarctic, which means that they start to undergo a death cycle. And the problem is that cardiac muscle itself is amitotic. It does not divide once it reaches full maturation and full size. Now, what's also interesting is that it is, just like smooth muscle, involuntary. However, it is autoregulatory and autorhythmic, which means that the cardiac muscle itself is going to be controlled and regulated based off of pulse rates coming from specialized cells within the heart. That specialized cell is referred to as the SA node, and it's that SA node that sets the heart rate and controls and regulates the heart rate. We do not have an actual centralized area of the cerebral cortex that controls the rhythmic rate of contraction of the heart. We do have neural innervation into the heart that can slow down and speed up the heart, but it does not actually regulate the heart. And so if we look at the cardiac muscle, notice that we have this kind of Y-shaped action, and notice that we have Ys interacting with each other where the prongs of the Y match up with the prongs of the Y at the intercalated disc. Notice we have that central nucleus and then that striation coming across. And so if we look at it in terms of the slide, the histology of it, notice how we have these kind of black lines running across the muscle tissue. Those black lines running across the muscle tissue themselves is the intercalated disc. The big purple spheres that you see on the tissue itself are not mitochondria like we saw with the skeletal muscle. That's the actual nucleus. And then notice how the striations are running in that banding pattern along the length of the muscle that we see here in the cardiac muscle. And once again, we'll get a little bit more detail on this as we look at the slides in closer detail in laboratory time.